Good morning, everybody, and thank you for the chairman and also to the participants for attending this seminar. First of all, I would like to thank also to the organizing committee for the invitation for me to present the opportunistic infection in Indonesia. So I would like to share the slide now. Okay. So when we talk about opportunistic parasite, so it is related to the immunocompromised status, and mostly for the HIV/AIDS patients, individuals. In Indonesia, the HIV prevalence is about 0.5% in general. It's quite small number. However, among a certain population, like the drug user, and among the men having sexual with men, the homosexual, the transgender, and also female sex worker, it is high. Like 29%, the highest proportion among the drug user, followed by the homosexual, and then the rest. And HIV patient with severely immunocompromised, they are prone to the opportunistic infection. Yeah, either that is from bacteria, virus, fungus, and also parasites. And it's already identified in Indonesia that 3 major opportunistic infection or co-infection among the HIV/AIDS. They are the tuberculosis, that's the number 1, followed by the oropharyngeal candidosis, and then the third one is those with diarrhea, covering up to 20%. Yeah. And then, of course, there are other opportunistic infection identified, but they are a very small number. And for the parasite, that is toxoplasmosis included in this. But the 20% of cases co-infection of HIV manifested as diarrhea, they are not fully identified. And at that time, in 2005, we don't know at all what is the prevalence of parasite among the HIV population. Yeah. So Indonesia is a tropical country which is endemic for parasite, and with its diversity of infecting human as well as animals. And it is already identified and reported that the helminth co-infection as a potential accelerator of the course of HIV disease, because it drives the Th2 polarization. So they are prone to the opportunistic infection. Yeah. And opportunistic infection, by definition, it is infection caused by the organism that's in normal immune competent host. It will not cause the disease. However, in immunocompromised or in immune deficient individuals, it will cause a fatal and severe morbidity. Yeah. So some of this opportunistic infection is considered as its defining illnesses. So that means if we able to identify this opportunistic parasite, then it is associated. There is probability, there is possibility that this individual suffer from HIV/AIDS or in other immunocompromised etiology. Yeah. So these are the opportunistic parasites, the Cryptosporidium species with the enteric manifestation, diarrhea, and then the Isospora, Microsporidia. These are all intestinal parasites with intestinal is disorder manifestation. Apart from that, Microsporidia can go for systemic infection as well. And then the fourth one is Toxoplasma gondii, which is a systemic and cause deep organ manifestation. And then before it was mentioned Strongyloides stercoralis as an opportunistic infection, but then WHO excluded. So the outline for this presentation, first of all, I would like to present the profile of intestinal parasites among the HIV in adult and children. And then secondly, the epidemiology of Cryptosporidium. And then the Cryptosporidium study in human. And the last one, that is the Toxoplasmosis epidemiology and updates, yeah. Because these two parasites, these two opportunistic parasites, they are the main parasites which affect worldwide population. So the intestinal parasite profile among the HIV individual, yeah. First of all, the study on the adult HIV patients with chronic diarrhea. Those study into hospitals in Jakarta, we check the stools from these patients with diarrhea by microscopic examination and performing also modified acid fast staining to detect the Immunococcidia, the Cryptosporidium, Isospora, and also Cyclospora, and also for gram chromotrope, we for detection of Microsporidia. We do also culture to detect Blastocystis hominis. And this is the result, yeah. So among the adults, we can identify high prevalence of intestinal parasite infection, reach up to 80%, yeah, more than 80%. And then single infection consists of 65 to 70%. And there is, yeah, polyparasitism among this population, yeah, either mixed with other protozoa or mixed with helminths. And then Blastocystis hominis, that is the most dominant parasite, yeah, found among these intestinal parasites, up to 70-80%, and then followed by the Cryptosporidium, yeah, about 12 to 17%, while Cyclospora we can identify in Cipto Mangunkusumo Hospital, but not in infection hospital, Sulianti Saroso, yeah, we cannot detect this. And then Isospora belli, there are very few, only one or only two cases of Isospora belli found during the study. And also Strongyloides, only very few, yeah, only one individual, if I'm not wrong, yeah, from this study. And this person actually co-infects with Isospora as well, yeah. And other pathogenic protozoa, Giardia and Amoeba, they are we can found, yeah, but in particular the Giardia up to nearly 2 to 3%, while in Amoeba is very low, and helminths not found. Yeah, only very scarce, yeah, helminth infection. And Microsporidia we can find at low number, 1.6%, but this only a proportion of the 348 individual, yeah. And so we can see here that in HIV patients, yeah, high intestinal parasites, yeah, and mainly they're dominated by Blastocystis, and Cryptosporidium is the second one found, yeah, mostly found. How about the profile of intestinal parasites among the children with HIV? So we compare, yeah, children with HIV in Cipto Mangunkusumo Hospital in Jakarta, and then we compare the profile of intestinal parasites to children in the population, yeah. And so there are 3 study sites, yeah. So among the HIV/AIDS patients, only small numbers, yeah, only 22 patients. We get the profile, yeah, similar like the adult patients, yeah, where the intestinal parasitic infection more than 70%, yeah, up to 80%. And then a single parasitic infection can cover up to 50 to 68%. And also polyparasitism is observed, not only among the HIV/AIDS, but also among the normal population. So the normal children population consists of those under 3 years old, and then the other one, those in primary school, yeah. And then the parasite, the main parasite was Blastocystis hominis in HIV patients and among children below 3 years old. However, among children, the primary school children, the blastocystis prevalence is not lower, yeah, much lower. And then the Cryptosporidium only found among the HIV patients and those children under 3 years old at the prevalence of 9 and 2%. So the prevalence of Cryptosporidium among the HIV is higher than the normal population. And other pathogenic parasites like Giardia and amoeba and also the worms, we did not find among the HIV. However, among the population in the community, we can find this one in the primary school, very high prevalence of STH, yeah. The Ascaris up to 60%, and then also Trichuris, half of it, and then hookworm. We can find those 3, yeah. This is in Jakarta. And then for the opportunistic infection, the Cryptosporidium, we did not find. So from this study, we can see the contrast between the HIV and the normal individual, where the Cryptosporidium is higher among the HIV, and then we can find among the children under 3 years old, but we did not find among the primary school children. In contrast, yeah, to the other pathogenic intestinal parasites, the STH is very high among the primary school children, yeah, and not found among the HIV, yeah, and also very few, yeah, among the children under 3 years old. So in this case, the protozoa is the main problem among the intestinal parasite. How about the CD4 level among the HIV/AIDS patients? Most of them under 100 cells per microliter, yeah. And then especially 50% of those with cryptosporidiosis have CD4 count below 50. So in conclusion, yeah, intestinal parasite is very high, yeah, among the HIV and also among the normal population, and mainly dominated by blastocystis, while the Cryptosporidium is the opportunistic parasite found in the HIV and children under 3 years old. And then we can see that fewer parasite species found in children infected with HIV than the adults. The Cryptosporidium prevalence, of course, it is higher among the immunocompromised and easier to find than in normal population. So we focus on the Cryptosporidium study, yeah. because Cryptosporidium is the opportunistic infection, and it is associated with the HIV/AIDS, especially those with low level of CD4. Yeah, so this Cryptosporidium, we know already that Cryptosporidium infection, it is an important zoonosis, yeah, because of causing diarrhea among the calf and cattle, and then it has many hosts. It is a zoonosis. Many reports on the among the animals, while among the children, it is prevalent for children under 3 years old. Yeah, it causes 20% of childhood diarrhea in developing countries. And yeah, so infection in early childhood will bring poor cognitive function and also failure to thrive. Yeah, and the specific of this Cryptosporidium, it doesn't inhabit in the intestine, but also can travel outside the intestine. Yeah, may trigger biliary disease, hepatitis, pancreatitis, and also arthritis, and possibly respiratory tract infection. So this is the life cycle of Cryptosporidium briefly. So this oocyst, yeah, if it is ingested or inhaled, yeah, it will excistate in the intestinal lumen, and then it will invade the enterocytes, and then inside the enterocytes, it will transfer into the trophozoite stage. Yeah, the sporozoite will then develop into the trophozoite. The trophozoite will develop into the meron type 1. This meron type 1 is like a schizoid. Yeah, it will explode and then produce other merozoite. This merozoite will undergo asexual reproduction, and then some will develop into the meron type 2, which will develop further into gamon or gametocyte. Yeah, macrogametocyte and microgametocyte will mate and produce sporocyst. This sporocyst can be either thin wall sporocyst that has capability for autoinfection, and then it is in small proportion, and or thick wall oocyst, which will be excreted through the defecation, and this is the type of resistance to the environment. Yeah, and important for the transmission. So the transmission can be person to person, especially among the household. When a person, household member is infected, and then among the homosexual, and also in daily care, yeah, in daycare for children, nursing children. Also, the transmission from animal to human is possible, waterborne, foodborne, and airborne. So up to date, there have been identified 20 Cryptosporidium species and genotypes. However, these species are found in human mainly. Yeah. How about in Indonesia? In Indonesia, yeah, there have been several studies or reports on cryptosporidiosis or Cryptosporidium infection. However, there are not so many, and then the studies are mainly among the animals. Yeah, and so this is the reports from Sumatra. Yeah, the green one, that is the study among the animals, but the red one, that is the study among human. Yeah, the human studies. So you can see here the Cryptosporidium distributed in this island, the Sumatra, Java, in Kalimantan. It's reported among the orang utan, and also Bali and Lombok. Yeah. So how big is the problem of Cryptosporidium infection ini or cryptosporidiosis? Already reported in the previous slide that the for children under 3 years old in Jakarta, we observed 2.1% by microscopic examination, while among the HIV/AIDS with diarrhea, we observed 11.9%. However, when we use fluorescence antibody test or the PCR, yeah, of actin S rRNA, we can detect higher infection among the both population, among the children under 3 years old and also among the HIV/AIDS with diarrhea. The difference is that if with the sensitive techniques we can detect at equal proportion, more or less, yeah, between immunocompetent and immunocompromised, but children under 3 years old can be regarded as not fully immunocompetent, yeah. However, the difference between microscopic and molecular technique or the fluorescent antibody technique, we observe that in children under 3 years old, by microscopic examination, it's difficult to find the oocysts. Well, it's easier to find the oocysts in HIV/AIDS with in HIV/AIDS patient with diarrhea. Yeah, so the intensity is different, the parasite density is different, makes it difficult. However, this is the actual prevalence. That is high prevalence of Cryptosporidium, actually, yeah, up to 33%. And then also another report among farmers in Yogyakarta. This is a thesis from our postgraduate student, yeah. So among the farmers in Yogyakarta, it's reported 13%, yeah, equal to the goat and also to the cow, the Cryptosporidium infection, yeah, using the PCR. And it's very difficult when performing the microscopic examination. So what species infecting human? What species of Cryptosporidium? So we perform the PCR RFLP of the actin S rRNA, yeah, using this technique validated by Xiao and also Nichols, yeah, and we can find, yeah, this is the procedure. After we isolate the oocysts, we do the DNA extraction, yeah, and then we do the PCR of the actin S rRNA gene at 2 loci, yeah, and digest it with this enzyme, yeah. And the result is that from 36 samples we check, that we find most of them, yeah, 3/4 with single infection of Cryptosporidium. That is the Cryptosporidium hominis, yeah, while 3 others with Cryptosporidium fellis and 2 with Cryptosporidium meleagridis. And then 1 double infection, Cryptosporidium hominis and meleagridis. And then 1 patient in 1 sample, yeah, we find 3 species of Cryptosporidium hominis, meleagridis, and also parfum, yeah. There are still some negative result, yeah, from this method, yeah. So among the children as well, from 11 samples we check, all we got Cryptosporidium hominis species, yeah. So we also do evaluation, yeah, of the treatment, whether the treatment given with paramomycin or azithromycin will affect to the Cryptosporidium detection, yeah. So those only 5 patients come for the post-treatment evaluation and check for the stool, yeah. So this one we check the stools using the microscopic auramine phenol staining, and also using the PCR RFLP to determine the species, yeah. And then these patients, some have reduced, but some has persistent, but some there is one, yeah, before treatment, yeah, before treatment, we cannot detect by acid fast staining. However, it is positive by PCR. And then after 3 months, this patient, yeah, infected with not only Cryptosporidium, but also Giardia and also blastocystis. So that means this may be a worsening condition for this patient, or maybe the behavior of this patient that she interact, yeah, she contaminated, infected with other protozoa, yeah. So before it was Giardia, but then followed by Cryptosporidium and blastocystis, yeah. So the Cryptosporidium species, they are identified here, mostly same, yeah, Cryptosporidium hominis. And then this one, this patient, which who before negative by microscopic and positive for C. miliagridis by PCR, on evaluation, follow-up treatment, harbor with 2 species of Cryptosporidium. Okay, but we didn't pursue further how this or why this patient got infected, yeah, with other species of Cryptosporidium. We did not pursue for the house and environment. And then apart from Cryptosporidium detection in the stool specimen, there is another report from Surabaya that among the TB patient, the tuberculosis patient that was admitted in Sutomo Hospital in Surabaya. So they checked the sputum and then they could identify 3 out of 44 sputum positive for Cryptosporidium oocyst. Yeah. And then the last one, that is the source of infection apart from human and also from the animals. And we check for the environment as well. So we examine the river. Yeah, there are reports on Cryptosporidium contaminated the river in Jakarta and also in Bandung. Yeah, in Jakarta, Ciliwung is the biggest river that becomes the source of clean water treatment process in Jakarta. Yeah, and in according to WHO, yeah, Cryptosporidium and Giardia is a parameter of clean and safe water. Yeah, on top of the coliform and other bacteria. And then examination of this river, we can find that there is viable Cryptosporidium oocyst, yeah, at this concentration, yeah. So these are the 6 samples. This one, yeah, we check using the acid fast staining and then auramin phenol staining, and then also using the immunofluorescence staining, antibody staining. Yeah, antibody monoclonal. And then using the acid fast staining, it's very hard and we did not find any positive result. But with auramin phenol, yeah, we can find there are 4 out of 7. Yeah, there are 4 out of 7 positive. While with the monoclonal antibody fluorescence staining, we can find even more. Yeah, 5 out of 7. So, and then to see whether it is alive or not, oocyst, so we stain with DAPI. Yeah. And then this is the enumeration, and then the highest one, that is the first sample that is before treatment. Yeah. And also apart from Ciliwung River, yeah, in Jakarta, there is also another report from Citarum River that is the source of water treatment for West Java. Yeah. And also there is another report of Cryptosporidium contamination in Karangasem district in Bali. Yeah. So this environment, the river become the source of contamination for Cryptosporidium. If human, maybe we can educate for prevention of the infection. How about the animals? Yeah, maybe among the animals, there need to be a strategy to prevent cryptosporidiosis among the animals. So Cryptosporidium, in conclusion, the Cryptosporidium is an important opportunistic parasite infection which causes diarrhea, yeah, among the HIV/AIDS and also among children, yeah. And it is the prevalence is high among the HIV/AIDS, yeah. Also among the children under 3 years old, the animals, and also we can find also the Cryptosporidium contaminated the river, as this becomes the source of infection. Human, animals, and also the rivers becomes the source of infection. And then the species Cryptosporidium found in human, identified in human in Indonesia, already identified C. hominis, Parvum, Liagridis, Felis, and Riley, where Riley is found in a traveler, among in a traveler from Australia, from Tasmania, in Bali, yeah. So C. riley, it is said to be, in fact, the Guinea pig, yeah. And then the species of Cryptosporidium reported among the animals already reported, that is Simiris, Parvum, and Andersoni, yeah. So the second opportunistic infection we studied, and also dominant one, that is the Toxoplasma. So when we see the global status of Toxoplasmosis or Toxoplasma seroprevalence in human, yeah, we can see here they are at different strata, yeah. The highest one, that is the dark red, and then the green one is the those with low prevalence, yeah. And then we see here in Indonesia, it is in the very in dark red, so very high prevalence, that is above 60%. So the Toxoplasma, when we talk about the Toxoplasma, we focus our attention, yeah, the impact of Toxoplasma for animals. Maybe it's not really a problem, but for human, it's a big problem, because it can cause a congenital toxoplasmosis for when in fact the mother, a pregnant mother. Also, it can cause severe manifestation for the HIV, because if main manifestation is to the brain, yeah, toxoplasma encephalitis or cerebral toxoplasmosis. But also to other organs, yeah, to eye, and causing ocular toxoplasmosis. And many individuals, they do not realize suffer from toxoplasmosis. They just complain of the blurred vision, yeah. And also systemic infection to the heart, to the kidney, yeah. So this is the vulnerable groups, yeah. The pregnant woman, fetus from the infected mother, immunocompromised, and those with ocular toxoplasmosis, yeah. The impact is high. So the organ damage depends on the host age, virulence, and also affected organ, yeah. If it, in fact, the brain and eye, of course, the daily, yeah, disability adjusted life years will be higher, yeah, and severe. So the brain disorder like this, it causes inflammation, brain abscess, not only single, but also multiple brain abscess, hydrocephalus, mental and motoric retardation, yeah. So the manifestation among the immunocompetent, generally benign or asymptomatic, some with lymphadenopathy, yeah, or express atypical symptoms like fever, fatigue, but some with visual impairment, but they don't realize that is due to toxoplasma, myocarditis, myositis, and hepatosplenomegaly. Yeah, and the characteristic of this of toxoplasma in immunocompetent individual that it cause asymptomatic latent infection. So as long as the immune system is competent, the immune system able to overcome the infection. So this parasite will hide in deep organ. Yeah, they encase in the cardiac, skeletal muscles, to the brain, in the brain parenchyma, and also in the retina. So, and it's already identified 3 types of Toxoplasma gondii, type 1, type 2, type 3, which is associated with the virulence. And type 1 is the most virulent one, and very potent and also easy to disseminate it. Yeah, and apart from these 3 types, there is also atypical type. Yeah, the virulent strains may also cause like severe pneumonia and disseminated disease. Like in Brazil, they have identified this type. Yeah. And among the the animals, yeah, Toxoplasma infection prevalence doesn't much change, yeah. From 1976 to 2090, a vet report, yeah, here that among the goat, yeah, and sheep, yeah, among the goat, 100% can reach up to 100%. Also in the sheep up to 82%, but among the cat, 41%. How about the cow and ox? Yeah, that is less among the ox, but among the cow, there has been reported 92%. Yeah, about the chicken and also the duck, yeah, it is less, yeah, less than the cat. And also among the pig, it is reported up to 75%. So there's still high prevalence of of Toxoplasma infection among the animals, yeah, the poultry, the cattle, yeah, and also the cat, yeah. And then this is the profile, yeah, of Toxoplasma infection in Indonesian population, yeah, from very old days in 1976, already reported up to 51% in Kalimantan, but in Sulawesi, the recent report in 2017, 58.5%. So it doesn't much change, yeah. And how about among the pregnant woman? The pregnant woman, yeah, the prevalence may be reported higher before, but now recent report in 2006, yeah, 27% among the fetal tissue, but up to 17% among the women with spontaneous abortion. And the ocular toxoplasmosis in old days, yeah, in 1980 report, yeah, 60% to 16 to up to 90%, yeah, among those manifested as chorioretinitis. So that means the clinical symptoms is very specific, yeah. So how about the toxoplasmosis updates, yeah, nowadays? So our study involving the Department of Neurology, yeah, the cerebral toxoplasmosis is high, yeah, among the HIV/AIDS. It is a problem among the HIV/AIDS and rates up to 33%. This is reported in Imran from among the neurology patients with cerebral disorder, yeah, in HIV patients with cerebral disorder in Cipto Mangunkusumo Hospital in Jakarta. And also similar study reported similar proportion, yeah, 30%, yeah, in Bandung. So this is the common manifestation, yeah, Toxoplasma encephalitis, and already identified that is because of reactivation of latent infection. And then the clinical manifestation may manifest as headache, seizure, focal neurology signs, fever, or maybe paresthesia. Yeah. And this is the imaging. Yeah, we can see the abscess, micro abscess, or the tumor. Yeah. And then this one is the big one, the bigger one. Yeah, distribution, not only one lesion but several lesions. And then so we check, yeah, the spinal fluid of those HIV patients, yeah, with cerebral disorders, yeah, and then from the spinal fluid we can detect, yeah, there is positive 50%, yeah, nearly 50%, yeah, positive for Toxoplasma IgG in the spinal fluid. And then when we check for the sensitivity and specificity, yeah, in comparison to the radiology, imaging, that is the MRI, then we get that the IgG, yeah, positive prediction value, that is 41%, but the negative predictive value, that is 71%. So we are not satisfied with this. We continue, yeah, and then we check the PCR of those patients, yeah, already detected, yeah, by IgG serology, and then using the PCR of B1 gene, yeah. And they are 58 cerebrospinal fluid, yeah, and then 49 already reported before. That is with the clinical diagnosis. If the clinical diagnosis consists of tuberculosis meningoencephalitis, yeah, that is the most, yeah, incidence, the most highly incidence, yeah, of cerebral disorders among the HIV/AIDS. And then secondly, the clinical diagnosis as Toxoplasma encephalitis, 28%, cryptococcal meningitis, 12%, and then non-diagnosed, yeah, for the rest, yeah. And then the nested PCR of the B1 gene showed 26%, 26%, yeah, positive for the PCR, but the IgG anti-toxoplasma, that is 28.6%. And there is a significant correlation, yeah, we found the correlation 0.5, and statistically it is significant. So we can confirm that the Toxoplasma encephalitis or cerebral toxoplasma among the HIV/AIDS is 28 or near, yeah, 30%, yeah. And similar study in Bandung, they use 64, yeah, they study on 64 spinal fluid, and also they for the PCR, yeah, they check the PCR Toxoplasma gondii using the PCR, yeah, real-time PCR in the spinal fluid, and they got 32.8%, while the IgG anti-toxoplasma in the serum positive for 77%, twice of the indicated by the PCR. So in conclusion, we can say that the prevalence of Toxoplasma encephalitis or cerebral toxoplasma among the HIV/AIDS in Indonesia basically about 30%. Yeah, it's quite high. How about the ocular toxoplasmosis? Yeah, the ocular toxoplasmosis, and this is a study on uveitis. Yeah, so the characteristic manifestation, retinochoroiditis. If the patient come with retinochoroiditis, which can be diagnosed by ophthalmology examination, then it's already confirmed this patient suffering from ocular toxoplasmosis. Yeah. However, yeah, there are patients come to the clinic with uveitis, yeah, and it's not significant or characteristic of uveitis, and we call it atypical infection, yeah, atypical uveitis. And then this is the study by Ladistia in Cipto Mangunkusumo Hospital in Jakarta among these uveitis, this atypical uveitis, yeah. Among the uveitis, 63% of infection, yeah. Uveitis can be due to infection or non-infection, yeah. And then 63% of the infection that is due to the toxoplasma, yeah. And then another report on ocular toxoplasmosis among the HIV/AIDS, that is 8.4%. And the uveitis manifests as chief complaint, that is blurred visions or floaters and so on, yeah. And uveitis is actually not only infection, but due to other inflammation. So it is actually inflammation of the uvea and the pigmented layer between the inner retina and the sclera and cornea. So it can be classified into anterior and posterior uveitis, yeah. So the problems, yeah, for this uveitis, yeah, for the blood vision, yeah, with this inflammation, that there is non-specific manifestation of ocular toxoplasmosis, yeah. So non-specific manifestation of uveitis, whether it is due to toxoplasma infection, and if so, what is the type of toxoplasma causing ocular toxoplasmosis, yeah. So this is the study, yeah, that is the etiology of uveitis of and 30%, 63%, 30 out of 48 patients, yeah, due to toxoplasmosis. And this is the result, yeah. Among those with atypical presentation of ocular toxoplasmosis, that is the atypical uveitis, we found that 12.5% toxoplasma, yeah, by PCR of B1 gene. And then we do the typing, yeah, and we found that it is the SAC2 type 3, yeah. So we cannot determine actually the type of the toxoplasma, type 1, type 2, type 3, because it's only based on one locus, that is the SAC2. We actually perform two loci, that is SAC2, that is the surface antigen, glycoprotein, and this one, the GRA6, that is the granular antigen, yeah. But this GRA6 didn't give any result, while the control show a positive result. So it's difficult to get amplification of the GRA6 locus using the clinical specimen, yeah. So the clinical specimen we got, that is the ocular fluid, either the vitreal fluid or the aqueous hummus, yeah. So this is the phylogenetic analysis, yeah, of those 1, 2, 3, 4, 5, yeah, 5 patients showing the type 3 of SAC2 locus, yeah. This amplified 3' and 5', and then this one as well, yeah. They are. We can conclude that based on the SAC2, the surface protein 2 typing, that is the type 3 toxoplasma infecting the atypical uveitis in Indonesia. So in conclusion, this toxoplasma infection are still high prevalent, yeah, and it's particular among animals, but among animals, even though it is asymptomatic, but this becomes the source of infection. It can become the source of infection to human, yeah. So we should be careful. And then toxoplasma infection in human may cause fatal pathology, yeah, among the HIV/AIDS due to the cerebral disorder, yeah, due to the reactivation. So it is very important, yeah, for the prevention. And Professor Somchai already presented before about the health education and then prevention strategy. Yeah, so the health education, because different culture has different type of education, but basically, yeah, using this a new era, yeah, communication, maybe we can spread information about this toxoplasma and also this opportunistic infection better, yeah, because parasite is already included in neglected tropical disease. So even though it is high prevalent like this, but the concern, yeah, awareness is very low, yeah, not only among the population, but among the health personnel as well. Yeah, so it needs an integrated management strategy involving the host agent and environment with multiple shareholders that it will be beneficial. And then, of course, yeah, there should be more study on toxoplasma, yeah, in human, yeah, and our interest, that is toxoplasma type determination. If we are able to use the clinical specimen, that is very good because it is associated with the manifestation, with the pathology directly, rather than we do the culture and then it's time consuming and so on. Yeah. Okay. So that's all about the presentation on the opportunistic parasites in Indonesia for the Cryptosporidium and Toxoplasma, and I would like to thank the contributors, because without them, there won't be information on Toxoplasma or opportunistic infections in immunocompromised in Indonesia. So we start actually in 2005, yeah, setting up the clinical parasitology laboratory with these personnels, and then supported by colleagues from Glasgow, the Scottish Parasite Diagnostic Laboratory, and with the mentor, the DCs, Professor Hugh Smith, and then later on we continue. Yeah, there have been many studies and reports on toxoplasma nowadays involving personals, yeah, in our department, and also the students here, and also support from Balitvet, that is the veterinary research institution, yeah, in Bogor, and also the neurology department, yeah. So thank you very much for your patient kind attention.
Thank you very much, Professor. Thank you for your very informative talk. This topic draws a very attention from the participants. There's a lot of questions in the chat box, but so sorry that we are run behind the schedule. So could Professor please respond the questions in the chat box?
Okay.
Could you please respond the question by chat in the chat box? So we will move to another session.
Okay. My question is how to isolate oocysts of Cryptosporidium from water samples, and what are the species of Cryptosporidium found in water?
Yeah. Yes.
Okay.
Please type in the reply in the chat box. Thank you so much, Professor.